So we're gonna go ahead and get started. Good evening, everyone, and welcome to a fantastic evening here with WSIM and AWIS and some of our other organizations. I saw the Wildlife Society has some members already joining in here tonight. Tonight is a very special night for us. We actually have one of APUS's own professors, Dr. Nate Bickford. He is going to be presenting a lecture this evening regarding habitat fragmentation and all things dealing with wildlife tonight. So we are super excited. I know I had Dr. Bickford a couple of semesters ago for general ecology, and I loved the course, and it was so amazing. So I'm really excited to have him here this evening to present to us, and I hope everybody loves all the information he has to share. Dr. Bickford, are you ready to take over?
Well, sure. So I have a question for you. It's not a big group, and we can take a vote. For some reason I thought we were talking about trapping wildlife. I can talk about wildlife. I can talk about habitat fragmentation or trapping and monitoring wildlife movement. They're similar topics because we use monitoring movement to look at habitat fragmentation. So I can do either one. It's kind of up to the group, I guess. Since we have a small group, we could kind of take a pseudo vote if you want.
I'm good with either or. Does anyone have a preference? Oh, well, let's see. Yep, there you go. All right, Dr. Bickford, whichever one you want to go, since you got a trapping wildlife already up, go ahead and we can just go roll with that one. That way you don't have to reset everything.
We've had this thing planned for so long. I think we've both forgot what. So, I'm a professor, of course, but I'm also the Colorado chapter of the Wildlife Society. I'm the president. I've been that for a while, so I've done a lot with wildlife and whatnot. My personal work is really looking at wildlife movement, and I've done that both wildlife and fisheries. So tracking where animals go and why, and what kind of habitat are they using, and part of that, and the story that I'm not going to present right now, but could I guess another time, is identifying the habitat that they're using and why is that habitat so important. And then we can use that to start managing habitat to create source habitats instead of sink habitat. So sink habitats are where a lot of biomass goes into that area, animals, and then they don't reproduce very well, or they get extirpated by predators or whatever. So that's a sink. And then a source habitat creates more biomass than it started with. And so we want to, because of habitat loss and habitat marginalization, you want to try to create as much good habitat as you can by managing it. Part of that is understanding what are the needs of the wildlife. And so I trap a lot of different critters and put transmitters on them, and then we can identify where they're going and what resources are they using. Right now I have somewhere in the 30s or so, or 40s transmitters on red-tailed hawks, goshawks, coyotes, bobcats, badgers, lagomorphs. My personal favorite is I love rabbits, so we do a lot with rabbits. But I've actually been doing this before I was even a professor. I'm a falconer, and I've been doing falconry for a very long time, and we trapped hawks, and so I knew how to trap hawks, and I kind of transitioned that into work as well. And this is my son and I with one of the goshawks that we have that we just caught a duck. But it doesn't go just there. This is a red-tailed hawk that we trapped. Here's a transmitter on its back, and the bird's got it's a backpack. It's a ribbon that goes around its body, and the bird's ready to be released after I get my my hero shot, right? And part of this was to show how it fits and where it goes, but it's always a fun picture to have. We do this specifically for the red-tailed hawks. We were looking at how they're migrating and what kind of resources do they need along these migratory pathways. And there's been some really interesting findings from this. We've actually identified populations that don't migrate at all. They stay on their territory all year long. And then individuals that migrate every year almost 4,000 or 5,000 miles and come back to the same locations each year. So it's pretty amazing data that we're getting. One of these transmitters, it's a solar powered transmitter, I've had on some red tails almost 5 to 6 years. So we can gather a ton of data. Most of these transmitters report every hour. And it really depends on what kind of transmitter you have, if that's reporting every 10 minutes or every hour or every 3 hours or what have you. And with solar power, we're lucky we don't have to worry about batteries, but a lot of this species that don't have that ability, they have to worry about batteries. And so the transmitters can get kind of large. With birds, you can only be 3% of their body weight. So you have to be really conscientious about how big a transmitter you can put. This transmitter is about 22 grams, which on a red tail hawk like this red tail hawk was about 1,000 grams. So we're well under our allowable weight for that. So, so anyways, we, these transmitters just give us a ton of information. I'll show you a little bit later, a couple figures of actual movement patterns, so you can kind of get an idea of what we do with that. So this was actually in Finland, and I was banding and collecting owls, boreal owls, and the mother was very, very mean and abusive, but the babies were cute and loving. And we weren't putting a backpack on this guy, but we did on the mom. So we caught the mom and put a backpack on her. And we banded the babies and put the babies back in, and away we went. But there's almost nothing cuter and uglier than a baby owl. So you got, every time you have to have a wildlife talk, you should have a baby owl picture in there. Here's actually a baby red tail, so it's not nearly as cute and really, really mad at me. One of my grad students had that climbed up the nest, pulled the baby down, and we put a backpack on this bird. This bird was trapped in Pueblo, or collected out of the nest in Pueblo, Colorado, backpacked, and then it went down and lived in Louisiana for about a year, and then came back to Colorado, and then went over to Texas. So, and so it'd be a 3-year-old bird now, and it's still kicking around and nesting and stuff. So it's, we got a lot of data out of this one baby, because we actually don't have a whole lot of data on birds coming from a nest, because frankly, a lot of them die. The mortality for a red-tailed hawk is about 95%. So 95% of all the babies out there just don't make it to the next year. It's hard to be a hawk in today's world. So we don't have a whole lot of data. We don't have a whole lot of data for most bird species out of the nesting season, because it's really hard to find them, track them, watch them. And so things like Christmas counts, the Christmas counts, you should get involved in, because those are really important data points that, that tell us a lot. And so any data that we can get that's not in the nesting season can be really useful. Now, I don't just trap hawks. This is one of my trapping crews, fantastic group of individuals. This is a badger. Now, I actually kind of like badgers. When you trap them, you don't have to chemically immobilize them. So we have this, like, if you take a dog or a cat to the vet and they have to do a surgery, they can knock them out, right? In wildlife world, we call it chemical immobilization. Badgers don't necessarily have to be. They actually don't have their talons or claws, are pretty thick. They're blunt because they're digging through the ground, and so they can't really scrape you, and they're really easy to scruff their neck. So badgers are pretty actually easy to deal with, but for safety's sake, we actually chemically immobilize the badger. Then we can put the transmitter on him and let the badger go. This was a small little male. It was really cute, and we got our quote unquote hero shot, which was really fun. Good group of folks to work with. Again, we were looking at badgers. Almost nothing has been done on badgers about, where they're moving, how they interact with other badgers. And the grad student on this is actually holding the clipboard, and he's got phenomenal data on 7 individuals that all inhabited a fairly small area. And their interactions are really quite interesting. And we can tell their interactions because the transmitter that we're using for this animal actually reports every 10 seconds. And so we get a ton of data. And so we can tell when individuals come into contact with each other, or even when they come into contact with prey, because the other thing that we had in this area, we had both coyotes and all the lagomorphs. So we had black-tailed jackrabbits and cottontail. We had transmitters on them reporting data very rapidly. And so we actually can look at the interaction. And you can use these interactions for a lot of different ways. Some of them for disease modeling. So how often do they come in contact with each other, especially with the rabbits for the hemorrhagic virus? We're using that to look at vectors, like how quickly do they interact, how often do they interact with each other, and then predator-prey interactions as well. This is a bobcat. Now, bobcats are not, I'm not actually holding its head down. Well, I am, but I'm not pressing the pole against its head. It's got what's called a catch pole. So it's got a noose around its neck, and I'm pushing that to the ground. So the pole is actually on the ground, but the noose is around the neck. The reason we do that is because bobcats like to wake up. So even with chemical immobilization, cats can be a little tricky, and in a lot of ways bobcats are the most dangerous for this, because of those the back claws can really carve you up a bit, and so you have to be really careful with bobcats. But bobcats are really interesting because they're kind of very generalist, and their interaction with other mesopredators is really kind of interesting, and their interactions between each other really gives you a lot of kind of interesting understanding of how these, what we call it, interguild. So that's interguild, like a guild would be a guild of predators. So how do bobcats and coyotes and badgers all interact in the same area? And we were looking at aspects of that. And because bobcats have such a big territory, their interguild kind of interactions change depending on how many are in that area at any given time. And so you have this ebb and flow between coyotes and bobcats in areas. It's really kind of neat behavior that we are identifying with movement. Caught another badger. This is one of my grad students who is unbelievable, best grad student I've ever had. She just took an amazing job in Washington. I'm super bummed. I would have liked to have failed her and kept her forever. She's amazing, but super good at managing all of this. And I'm telling you, badgers might be the cutest, misunderstood little animal ever. They're just super neat little boogers for sure. This is, so you can see those claws. I always want to say talons because I'm a bird person, but the claws are really kind of blunt and really actually easy to deal with. We keep their eyes covered. I should have mentioned that, so the light doesn't stimulate and get their adrenaline going, because they're just kind of somewhat knocked out depending on what level of interaction, and then they come out of it pretty quickly. There's those claws. They're big though. They are definitely big claws, and super cute. They look flattened, but they're not flat completely. And I think they're adorable. So this is one of my undergrads. Definitely have a lot of undergrads doing this work and working with us. This is one of those 7 badgers that were caught. And I should have mentioned that towel is actually there. We put that in on a scale that has a hook on it, and so we can measure the weight of each of the animals. And all of these animals got transmitters released and are still doing quite well. So this is a coyote. When coyotes are tricky, they are really hard to trap. So we actually trap them in foothold traps. Now I know everyone's like, oh my god, the foothold traps with the big claws and all that kind of stuff. No, these are padded. They're actually pretty easy on the animals. We haven't seen any problem at all with them, at least in our study. So we've been very lucky, I think. But when they start to come out, so this coyote, we gave them the drugs to wake them up after we gave them the chemical immobilization, and we just kind of keep our eye on them. We watch them, make sure they get up and walk away safely and healthy and all that kind of stuff. And there's the coyote kind of walking away, and you can see on its neck the transmitter. It's got a little collar, and yeah, that coyote was doing really well. We caught one the next day that made it about 6 months and then was found dead on the road. We think probably shot, but we got a lot of data beforehand. Here's another. This is a pretty big bobcat, and this bobcat did wake up. So as we were getting the collar on, it was you can see the collar there. it was starting to wake up at this moment, and I was reaching down to pick it up to make sure that we had all of our data collected. This is one of the lagomorphs, a bunny rabbit. This is a desert cottontail. We have mountain cottontail, desert cottontail, and white-tailed jackrabbits that we have in our study. And again, put collars on it, looking at movement. And one of the things that this study is that I was really interested in, is we weren't just studying one species. We were really looking at the whole trophic cascade. We had deer, rabbit, predators, so the whole gambit. And it was really kind of interesting. Really hard to catch cottontail rabbit, believe it or not, mostly because we had so many kangaroo rats, and they would come in, eat all of our food, and then leave. So they have these big pouches, and they come in and get all the seeds in their big pouches and leave the trap. Even if they not made the trap go, they could fit through the holes. They were such a pain, but we did catch some, and it was really great data. We got amazing data for rabbits, and really looking at where are they moving, what resources do they need. And this is really important because actually lagomorph numbers across the country are decreasing. So there's a lot of places where these guys are really important and really lacking on the landscape. You got to think, like, cottontail is one of the most important prey items for most species out there, predator species. And so I call them the nature's Snickers bar, right? Everyone wants a Snickers bar. And if you don't have Snickers, you're very sad. And so these guys are pretty important. This is not a great picture, but if you understood, it's at night. So your camera phones aren't that great at it, but this is when we trap jackrabbits. So we go out with spotlights, and we find the jackrabbit, we surround it, and we come in. And as we get closer, they have the kind of the deer in headlights kind of behavior, and we can net them. It is a lot of chasing through desert and cactus with nets, and everyone gets a lot of cactus in their body, and it's pretty fun. It's like a big fun goat rope, but it's a great way to trap jackrabbits. We've got a paper coming out about the best methods to do this and whatnot, but it's pretty neat to get jackrabbits and put collars on them. And here's maybe a little bit better picture. We've got all of our sampling gear here. We've got everything in our sampling gear, from the nets, but also if an animal gets injured, like if a rabbit gets a cut in its arm or something on a cactus, we'll sew it up, put antibiotic ointment on there, all that kind of stuff. We'll just make sure that they're okay or whatever. Luckily we haven't had any real issues or anything like that, but we're prepared for all of that. We have cleaning material to make sure everything's clean. We use pillowcases to keep them in while we're getting the collar on, things like that, to keep them calm. We do everything we possibly can to keep the animal calm and safe. And that's one of the reasons in the previous picture, they're using the red lights, because red lights, wildlife don't react to them quite like they do white lights. And so they're calmer and less stressed. So let's look at some data. This is actually, each dot is a point in the movement of these are jackrabbits, and believe it or not, this is like 3 weeks worth of data. So it's not a lot of data. It's a lot of data for a very short period of time, but because we collect so much data, we can saturate our understanding of an area. And so you can see that there's some distinct territoriality within these jackrabbits, and yeah, a ton of data. It's really neat. And you got this one in purple that's using this kind of drainage area. You've got the light green and the blue, and they're distinctly different. And it's not terribly surprising. These are all males. These happen to be males. And so they're not that friendly with each other. The females tend to overlap quite a bit more. So, but it's amazing data for sure. This is one of our camera captures. This was a swift fox that got one of our rabbits in its mouth. I wasn't super happy about the rabbit getting killed, but we got the data. We got the actual whole hunt and chase and catch on our telemetry, or so on the collars. So it was really pretty amazing data with a camera corroboration. So along with all of our movement data, we also have cameras out there that's capturing a lot of data. Now I'm going to show you a species we do not have colored. So we also have mountain lion down there. And although my grad students were very mad at me that I did not want to trap cougars, I've done it before. It's not really that much fun for me. And I didn't want to deal with having all that you have to deal with mountain lions. So, but there's all kinds of interesting predators in this area. So here's some more data. This is actually the badger data. And so badgers, this is some of those data points I was talking about. What's interesting is you have some overlap and then you have, really distinct boundaries. And this is a mix of male and female. So why there's certain boundaries and not is kind of that interesting component. And we're still trying to figure some stuff out, but again, pretty amazing data. We also had transmitters on prairie dogs. And so you can see the little collar, and the prairie dogs are awfully cute. They're really hard to deal with though, because they like to groom each other. So they chew the collars off each other given time, even through metal. So they're pretty impressive. Here's some, these are prairie dog home ranges. So we can get, what kind of home range, where do they spend the bulk of their time, things like that. This is a home range for a rabbit over an 8-day period. And you can see there's actually a, well, you don't know, but the big spot is night. The small spot is during the day. So during the day, it actually is hanging out in a slightly different spot than where it does at night for the most part. So kind of neat interactions there. So all of this trapping and transmitter work comes with, lots of time in the field, which is kind of the fun stuff. And then you have these millions of data points that you have to deal with. And so if you're interested in anything like this, one of the most important things you can learn is R. So almost everything we do is done in R. Our mapping, our data analysis, everything is done in R. And there's various packages. So my students are working on things like home ranges, resource selection, social network analysis, things like that. And those are all done with using R packages. And if you have not heard of R, well, you haven't been, I think that one of the stats classes, as far as APUS, uses R, but it's one of those things that most of us have had to self-teach anyways. It's free software out there, and so we all have to learn how to do it as well. We use QGIS as well a lot, which is also a free mapping software. So a lot of things that are going on, at least in my lab, and with a lot of people I work with, is with R, which is free, and QGIS. And so self-teach. If there's no classes in school, there's workshops and other things like the Wildlife Society, which is in November, has an R workshop. A lot of people will go to those kind of things. Each state has a Wildlife Society chapter, and usually at the annual meetings, there's workshops. Ours almost always held. I know the Colorado chapter is having an R workshop during their meeting. So there's lots of opportunities to learn the tools and techniques that you need to know. What I typically find when I get grad students who have been out in the field, so I usually put an ad out at the Texas A&M website, and I get applications, and I usually hire somebody who's been out in the field 3 or 4 years doing tech work, and they know how to do the field work, and then we have to work on how to do data analysis and whatnot. If you come, if you apply and you already know how to do the data analysis because you did some self-teaching, and you know how to do the field work, it's going to set you up to be very successful. People will want you. I often call that the unicorn. Unicorns are hard to find, right? And the unicorn for wildlife are folks that know how to do the field work, but also know how to do data analysis. And so this is a good time to prep yourself for data analysis. Take the classes that APUS has to offer in statistics and data analysis, even if they're outside your plan, because it is the best way to arm yourself for successful job applications. So yeah, so we handle a lot of data and the camera pictures as well. Like often with cameras, 50 array of 50 cameras will produce about 1 million photos. And we use machine learning in Python and R to run through those pictures. We don't do any programming in R, but there's other people that, or excuse me, in Python, there's other people that do, and they've created these machine learning packages that will go through your pictures and tell you like, that's just grass. And so they'll throw those away. So you go from like 1 million pictures to like 200,000, and then you can start going through them, which is a lot easier to go through, but it is definitely a lot of data analysis and processing too. So you go from field to computer, field to computer, field to computer. So, I want to give time for questions. And so I didn't want to talk more than 30 minutes because I don't even want to hear myself for more than that. So I figure you guys don't want to either. So I guess I'll open it up for any questions.
Awesome. Thank you, Dr. Bickford. I actually had a couple of questions myself. Kind of going back to where you were mentioning the nesting season for red-tailed hawks. You had mentioned that there was a 95% mortality rate for the youngsters. Is that due to predation between like older red-tailed hawks or disease, or what is the reason for such the high rate?
To quote Shelley, it's kind of dealer's choice sometimes. Mortality from other raptors, typically maybe eagles and great horned owls are pretty rough on red-tailed hawks, but it's getting hit by cars, running into windows, electrocution. Electrocution's a real bad one. Getting shot. I had a couple different projects with grad students looking at humans shooting raptors. It happens a lot. There's a lot of raptors that are shot every year. So I mean, and red-tailed hawks are always sitting up on posts. And the other part of it is young raptors are terrible hunters. they make a lot of mistakes, and if they don't have a plentiful food resources, then they can only miss so many times before they're too weak to really hunt effectively. So
now I know I've seen like the eagle cams where like the parents stay with the eaglets for quite some time. Is that the case with the red-tailed hawks, or?
To some degree. I mean, there's some, all of the broadwings are kind of the hawks, if you will. There's some parent protection. I think red tails may be a little bit, longer than some other hawks, and they'll do some interesting things. I mean, I'm a little more familiar with that kind of stuff with goshawks. Goshawks parents will bring wounded animals into the nest area, and the babies have to chase them down and stuff like that. There may be some of that with red-tailed hawks, but for the most part, they're there to give them food. The babies will follow the parents around and beg for food for a period of time, and then the parents get really annoyed and chase them off usually. So
I wish I could do that with my child sometimes. Get annoyed and chase them off.
When they get older, they don't want to be around you anyways, so it's okay.
Exactly. Does anyone else have any questions? Yes.
All right, well, sounds perfect.
I think Ashley is probably handling her little nestling. Yeah. Yes. So in the few minutes, do you happen to have one of your birds handy that you could show us? Or they
don't
are they
actually?
not?
I'm in my office, so they're at home. So no, they're in the home, and my Mew happy not to be around me right now. So
fair enough. All right.
Sorry about that. Had to
Worse.
get out. But thank you. Let's see. I'm trying to see. Oh, for the R software that you were mentioning, could you possibly send us a link that I could include with the recording? That way anybody who wants to look at it, they can actually go ahead and start teaching themselves and exploring that as well, since you mentioned it.
Yes, I mean, you can Google it for sure, and there's lots and lots of free workshops out there. There's workshops you can pay for as well. I mean, Shelley, I'm almost positive that APUS has classes in it, or at least the math department, I think, teaches some. So I'm almost positive.
Thank you so much. That's awesome. This was so much fun tonight. I mean, I really appreciate everything. And like I said, I enjoyed taking your class. It was so interesting.
I think Ashley's being very nice, but that's okay. I appreciate it.
No, I did. I really liked your class. I had to make that, keep muting. I had to make that little ecosystem in a jar thing. And it was so cool because mine actually survived the whole 8 weeks.
Actually, one of my favorite projects for online learning is that. That's fun. That is a fun thing.
So, but yeah, no, I loved it. It was great.
Can you tell us more about that assignment, if you don't mind?
Me or Ashley?
Either one. She's laughing, I think. I don't know. I think she's handing you the mic, Nate.
Well, for ecology, which is not a graduate level class, but it's a neat assignment. You have to build a biome in a jar, and the idea behind it is it's an environment that can survive on its own, right? You don't add any more water, you don't add any more food, and so it can just keep going around. And I have one at my office at home that's a shrimp, and it's a ball. You can actually buy them. I made mine, but it has I put a little bit of algae in there, and the algae grows. The shrimp eat the algae, and then they go to the bathroom. The algae grows off the nutrients, yada yada yada. So it's a contained ecosystem, and you can build some very effective that actually do a really, really good job. And I'd say actually in the class about 75% of the students actually have living things. Not all of it's pretty living things, but usually it's living. But you get a lot of information from it. It's a pretty slick little assignment. I like it. And it goes through the whole course. You follow it. So.
Very cool. Maybe I can tap your brain a little bit about that.
Sure. Happy to do it.
Can you put your email in the chat, and I'll email you. Also,
Sure.
one other thing. Thanks, Nate. One other thing I've noticed, Sky on this Zoom call. Sky, are you a falconer? It looks like you're holding a raptor of some kind.
Hi, yeah. So I'm actually not a falconer yet. I'd love to get into it more. I volunteer at a birds of prey sanctuary in upstate New York. And what we do is we kind of rescue birds that would otherwise be euthanized. And we take them into a program where we educate the public on the birds of prey, their relationship, and kind of just basically working with a lot of people on just informing them about the raptors and everything. The specific bird in the picture is actually a really interesting bird. She's one of my favorite to work with. Her name is Rosalie. She's actually a Saker gear falcon. So interesting because you'll never find her in the wild. She's actually a man-made bird. She's a hybrid, and she basically is a cross between a Saker and a gear falcon. And she was actually a working bird in upstate New York at a landfill, and her job was to chase away nuisance animals from the landfill. She ended up in the program because she, for some reasons unknown, was electrocuted. So she flew into power lines and was permanently injured. And because she was already a bird that was used to being handled, they decided to, instead of euthanizing her, to rehab her, and she ended up in a program where we use her for education.
That's the coolest. Thanks for sharing that, Sky.
Yeah, absolutely.
Awesome.
Well, thanks guys for having me.
Thank you, Dr. Bickford, for joining and sharing all that information. For everyone who wasn't able to make it on tonight, this is obviously was recorded for us. So if anyone wants to rewatch this and get any more information, visit the links that Dr. Bickford shared with us. It will be up on the WSTEM YouTube page shortly. So you guys can definitely check it out there. Other than that, thank you again, Dr. Bickford, and everyone who joined us. I hope everyone has a fantastic evening.
Thanks, guys.
Good night, everyone.
Thank you, and have a good night.
Bye.